In this video, we're going to talk about prokaryotes. We've already talked about them a fair amount in this class, so we're not going to go real in-depth into some of the areas we've already covered.
Prokaryotes in section 8.1 if you're interested. So you already know that there are two kinds of prokaryotes. They take up two of the three domains of life, where of course the third one is eukaryotes, and all eukaryotes would fall into the eukaryote domain. The two domains that we're concerned about today are bacteria and archaea. Remember that bacteria are very diverse. They have peptidoglycan in their cell walls, and archaea typically don't cause disease, so we're not going to focus on them much today. They lack that peptidoglycan, and we classify them based on the kind of environment they live in. So they could be thermophiles, halophiles, or methanogens. The thermoacidophiles live in very hot, acidic places, so deep hydrothermal vents, sulfur pools, geothermal vents, etc. Then we have haliophiles that live in very, very salty conditions. Normally, osmosis would make salty conditions too hard for most cells, but these are organisms that can survive in these incredibly salty environments. And then finally, methanogens live where there's very, very little oxygen. So inside of intestinal tracts, in cows, and frankly even humans, they live in the really stinky lake sediment and swamps and things like that.
We've already studied prokaryotic structure, so just take a second and label it in the diagram. As we go into the next section, these are review as well, but since they're specific to prokaryotes, it's probably a good idea to write their function next to their label.
What is a little bit different is all cells have DNA, but the DNA of eukaryotes is divided into chromosomes, and they're paired chromosomes, as you know, one from mom, one from dad, etc. In prokaryotes, they don't divide sexually, and so they have just one circular chromosome that contains most of their genes. But they also have a bunch of little chromosomes, and this is drawn exaggerated to show it, but their little chromosomes are called plasmids, and those are the bits that are exchanged to increase genetic diversity. We'll talk more about that in a minute. The pilus. So a pilus is this guy here, and the pilus looks a little bit like cilia. Bacteria have those as well, but instead the pilus serves as a tube. So that tube can serve kind of like the arm extension on your vacuum cleaner, and that it can suck things up. It can also connect to another pilus, and the two pili can create a tube through which plasmids can be transferred. So that pilus is incredibly important when it comes to increasing the genetic diversity. We will talk more about that in a minute. Finally, in addition to having a cell wall, most prokaryotes have a capsule. Capsules are made up of polysaccharides. It's not necessarily a guarantee that all will have it. The capsule does things like help prevent it drying out, helps it attach to the surfaces that they live on, and makes it tougher to kill. Let's pause here to review. So there are some important characteristics of prokaryotes that we should discuss. The first of which is shape and arrangement. So we have three words to describe the shape of the bacterial cell. Some are spherical, so we call them cocci or coccyx. Some are rod-shaped, which we call bacilli or bacillus. And finally, some are spherical, so we call them spirilla. Additionally, you might have heard of things like streptococcus. That means, if it's streptococcus, that the bacteria are arranged, they live together in groups that are in twisted chains, and they have spherical shape. Or staphylobacillus would mean that they live together in clusters, but that they have rod shape. So that often tells us not only the shape, the arrangement, but that's also how we name a lot of our bacteria. So if we take a look here, in this very basic drawing, we have cocci, we have bacilli, we have spirilla, we have some that are in twisted chains, so strepto, we have some that are in clusters, so staphylo, and some that are in pairs, so diplo. Take a second before we go on and fill in the chart in your notes. I'm going to put the answer key up in a second, but I want you to try doing it on your own. As promised, here's the answer key. Double check your work. You may notice that we didn't talk about spherocytes or vibrios. I'm not going to ask that you know that. Let's pause here to review. If you've ever gone to the doctor and they've taken a culture when they think you might have strep throat, they take that long Q-tip and they take that swab is what they call it. They rub it on where they think the bacteria is living, and then what they do, and you don't see, is they go back in their office and they do what we call a gram stain. So the gram stain has a whole bunch of different steps to it. They take the Q-tip and they put it on a slide, then they heat fix it, and then they treat it with these different chemicals. When they're all done, the bacteria that's there will either be purple or pink. That purple or pink tells them about the cell wall composition. So the chemicals used in the gram stain respond to the amount of peptidoglycan there is in the cell wall. That's really important because some antibiotics work by breaking down the cell wall, the peptidoglycan. So by knowing that something is either gram-negative or gram-positive, they know what kind of antibiotic to use. So the two results, gram-negative bacteria look pink. They didn't hold on to as much stain. The reason why is because they have an outer membrane on them. And that outer membrane protects the layer of peptidoglycan. So if you're taking an antibiotic that works by trying to break down that peptidoglycan, it's not going to work as well because that extra layer is providing protection or barrier to the peptidoglycan. If you have gram-positive bacteria, it tends to look purple. That they have much more peptidoglycan without that extra outer membrane, so antibiotics can get to it much more easily. You will need to know the difference between gram-negative and gram-positive. I don't care so much that you know the color. What I care about is that you know that negative means less peptidoglycan, an extra membrane, and they're harder to kill. Think about it being a negative result. That's bad news. Or gram-positive, it's good news. Hey, there's antibiotics, you can treat it. More peptidoglycan, easier to kill. Let's pause here to review. We can also classify bacteria based on their metabolism. Remember, aerobic respiration requires oxygen, so we can call them an obligate aerobe. We can also call them anaerobic, meaning they're able to reproduce without oxygen, or facultative anaerobes, which means they can survive with or without oxygen. Let's pause here to review. Bacterial reproduction is also a review. We've covered it a number of times. Bacterial reproduction is very, very quick in ideal conditions, and it's through asexual reproduction. We call it binary fission. So binary fission happens in 3-4 steps depending on how you divide it. The first of which is the chromosome. Remember, they only have one. The chromosome is copied. Each of the two new chromosomes attached to the cell membrane, and then the cell divides in half by building a new cell wall between them. Again, we call that binary fission. It's very, very fast in ideal conditions. It is as quick as 20 minutes. And it's asexual. And because it's asexual, remember, that means that both of the cells are identical to each other. So that leads to very little genetic variation. That is, there are lots of ways that they can introduce new genes to their gene pool. So one of those ways is through conjugation. Conjugation is basically cell-to-cell transfer. Think of co meaning together. So it's basically two cells hook up their pili. So one's got a pili, the other one has a pili, and they make a little tube. In doing so, they transfer part of the plasmid through the pili, and each side is able to copy and make the second half of the DNA. They send one half through, they keep the other half, and they both get a copy of the plasmid. That's one way that they can increase genetic variation because it allows them to share genes. It's not really sexual reproduction, it's just increasing genetic variation. Another way that bacteria can increase their genetic information is transformation. Remember when we talked about Griffith and how the bacteria were able to take up genetic information from their environment and get that extra smooth coat from the dead bacteria? Well, that was transformation. They take up DNA from their surroundings. In this case, the pili act sort of like vacuums. You know, that tube extension you have in your vacuum, and it sucks up the DNA, and then it's able to incorporate it into its DNA. Usually it's from dead bacteria around it, but bacteria are known to pick up DNA from other organisms as well. There's also one called transduction. It involves using viruses to transfer DNA. We're not going to talk about that right now. Let's pause here to review. It turns out bacteria are really, really important. They have very important ecological roles to play, and without bacteria, we would not be able to survive. So there are a couple of really, really important things that you should know. Number one, bacteria are crucial to biogeochemical cycles. As we talked about, they're saprotrophs. In other words, they are decomposers. That's an important function they serve in our environment. The most important thing that you should remember from both from first semester and now is that we have chemioautotrophs that are nitrogen fixers. Remember, that means that they take nitrogen from our atmosphere and in either water or soil, they fix it into other kinds of nitrogen that then can be introduced to the rest of the food chain. So only bacteria do that. Not all bacteria, of course, but only nitrogen fixers, and that is incredibly important. You've probably also seen some commercials, or maybe you yourself take a probiotic. That's because we have bacteria that are what we call normal flora. In other words, bacteria that live in and on us that are completely harmless. In fact, they are very helpful. They aid in our immune system. They help compete with bad bacteria. They help us digest food. You've probably heard of Activia that's supposed to make your stomach digestion and whatnot better. There's been research to suggest that it has to do with your other digestive process, the amount of weight you hold, inflammation in your body, all kinds of really, really helpful things that bacteria that live on us help us do. I've also read that by number of cells, we have more normal flora bacteria in and on us than actual our cells. And that's a very good thing. Let's pause here to review. Bacteria, in addition to natural things like normal flora and their ecological role, have a tremendous amount of uses that we put them to work in recent years. So we use bacteria for food production that helps us make cheese, yogurt. Their fermentation is important for that. Chocolate, of course, making vitamin B12, all very, very important in it. We use bacteria extensively in genetic engineering, as you've already learned. So we use it to make recombinant DNA so we can make products like insulin that humans need. We use it for CRISPR-Cas9. That's how we discovered it. CRISPR-Cas9 is bacteria's defense against invaders since they don't have an immune system. They have CRISPR-Cas9. So very, very important for genetic engineering. We use it to do what we call bioremediation, which means that using one organism basically to solve an environmental hazard. In this case, there are bacteria that eat oil, bacteria that eat tar, coal, etc. And so when we have something that we want we want gone, if we can find a bacteria that eat it, then problem solved. Not entirely, but so for example, in oil spills, oil gets into a lot of places that we can't scoop up or vacuum up or whatnot, and so a lot of times we'll spray oiling bacteria on it to try to get rid of it. It's not foolproof. It's not, as I said, problem solved, but it is a really helpful tool. We also put bacteria in our sewage treatment plants to help eat sewage because there are a lot of bacteria that are decomposers and saprotrophs that love to eat.
That's it for this video. Later we're going to talk about the different diseases caused by bacteria and other disease-causing agents.